Thank you. Thank you for our great pleasure in capabilities Professor Dipjyoti IISER Kolkata for our department Colloquium today. They operate in the BSc and certificates in physics from Development University and ladies PhD in from IIT Bombay. After that, experience of postdoctoral solutions abroad, which is drawing IISER Kolkata and associate professor there. He works in a very interesting area, cross section of theoretical, static, condensed matter, and biological physics, because quite hard this interface. So we are all here to hear from you. Thank you. Thank you so much. Thank you for this opportunity to Mike. I think Mike is not. Are you all of you can hear right at the end? Because we all teacher purpose, so we are habituated to speak very loud. So we don't need mic. So the purpose of the talk is to introduce you in the area of what we call biophysics or the physics of living matter. Okay, and also give some exciting stories of our work in our lab. And the talk will be like a discussion. So feel free to interrupt me and stop me at any time. It will be especially to the students. By the way, why you are sitting at the back? Some of you can come in front if you want. So if you have any questions, stop me. Okay? And at least at the end of our talk or our discussion, you should be at least able to understand every slide that I am showing here. So that is the purpose. Okay. So first of all, as you can see, I am associated with biological sciences as well as physical sciences. It is a very problematic and also exciting position. So then what we do there to get that, let's finish. Let me take the background. Okay.
Next is a loser.
Loser.
ঠিক আছে loser.
Sweet.
তোর এটা কেন সমস্যা করছে কেন একটু সাইডের দিকে করে দিও হ্যাঁ প্রজেক্ট ওটা ইয়ে ছিল ওটা লোক বসে গেছিল
Okay. So as I told you, please stop me and ask questions, and you can also ask questions in Bengali, by the way, if you want. So this is our campus at IISER Kolkata. It's very far away from the hassle of this city. Quite a clean and nice campus. I like it there. It's like 2.5 hours by car from here. So first of all, what is the motivation of our lab, or in general, what is the motivation of biophysics? So if you ask a biologist, they will give you different definitions of what is life. And nobody understands. There is no answer to this question of what is life. If you ask molecular biologists, they will give a different answer. If you ask evolutionary biologists, they will give another answer. Okay? So for physics background persons like us, what is most exciting that there are two fundamental things about life which is very interesting. One is the diversity of life form. So you can see from this tiny bacteria with lot of this tail-like structure. These are called flagella. Okay, and from ant to the biggest mammals, you also have plants that cannot move but still responds very nicely to even like 1 or 2 degree changes in temperature. Okay, so the first thing that you notice is the huge diversity. And second thing that you notice in biology is that life kind of organized into different scales, different length and time scales. So it is like what we call in biology organizational scales. Okay, so you start from like molecules, like water molecule. Okay, at the level of Armstrong. From there you have different biological molecules, what we call macromolecules, which are like biological polymers. So this is a picture of a DNA. From there you can form a cell. Cell has different structures. Many cells come together, form a tissue, and then you also can go to what we call an organism, right? So this is a picture of a geographis embryo. Yeah, geographis is a tiny fish that you can find even in our Ganges. So a lot of people do experiments on geographis. So these arrows, you see, I am putting these arrows in both ways. What does it signify is that so there are crosstalks between the scales. Okay, so whatever happening in a higher scale cannot influence at a lower scale, and vice versa. And this makes biology is very exciting from the point of view of what we call statistical physics. Why? First of all, I told you two things: diversity of life and organizational, hierarchy of life. So these words like diversity, or Darwin used the word called variation when he described, the evolution in the origin of species, or species talk about the word heterogeneity. These are like different types of what we call statistical noises. So we use the tools of stochastic processes or random processes to understand that. Okay? Guys, better to switch down that fan. It was making that noise. Thank you. So, second is that we ask like how the macroscopic properties, like property of a tissue or property of a cell, how it depends on from the microscopic property of a lower scale. So if I know the properties of a molecule, can I say that how things are organized in a cell? Or if I know the property of a cell, how the collective mechanical behavior of a tissue will be, right? So these here you understand the framework of statistical mechanics is very helpful, because conceptually all of you have done this exercise of what we call kinetic theory of gases. So what we do there in kinetic theory of gases, we have some very simple rules of point particles. For example, we assume something very simple, like there are binary collision. There is no such thing that there is a three-body collision, right? So there are many simplistic assumptions we make, and on basis of that, we calculate or predict something at a larger scale. We calculate, say, the pressure of a gas, or thermal properties of a gas, and so on, conductivity and so on, right? So from microscopic assumption, we try to understand what is the macroscopic property, right? So these two ideas, talking about heterogeneity of noise and talking about behavior of macroscope from microscope, this really fits with the idea or concepts of statistical physics. So that is one of the very primary tool when we address biological problems. Statistical physics, both equilibrium and non-equilibrium or stochastic processes, will be a fundamental theoretical tool to address that. Okay. Now this is just a motivation, but then all of you must be kind of wondering that what is biophysics. Now it is very hard to define what is biophysics. So I am not going to even answer that. Rather, I am asking the question: who are the biophysicists? Okay, so whom we call as a biophysicists, and you see there are plenty of names even from the Indian context. So very early name, of course, J.C. Bose, and this is like all of you have seen this picture of his instrument called Crescograph, which he made to see the growth of plant, to measure the growth of plant very precisely in response to environmental stimuli, right? So this is one of the earliest example in Indian physics, what we call biophysics based instrumentation, right? So this was the completely homemade or made by a native person in the when we were still under the British rule, right? Now, if I extend the horizon, I have given you some names: Francis Crick, Linus Pauling, Ramachandran of our own, then Alan Turing, Max Delbruck, John Hopfield. So if you see all of them, we categorize as biophysicists. If you go to the Wikipedia page, you search this name, you will see that they are categorized as biophysicists. And I am showing them in two different colors. Why is that? Because if you look at the subject matter they are studying is hugely diversified and different. And what is that? It's very intuitive because life is so diverse. The first point I told you is that there is no way you can, address what is life without addressing the question that how diversity comes in life. So, of course, when we are studying what we call biophysics, there is a diverse school that all you can call the umbrella of biophysics. So, this, scientists in this red color, their main motto is to understand the properties of what we call macromolecules. You see that why they are famous to understand DNA structure of hemoglobin, protein conformations. So these are all structures and conformations of biological polymers or macromolecules, right? And people have used the ideas of quantum mechanics, even classical mechanics, to understand the structures. So this is one branch of area what we call biophysicists. Another set of biophysicists, they work on a very diverse topic. Alan Turing, one of the famous contributions is to understand what we call morphogenesis. What is this word of morphogenesis? Many of you may be hearing it for the first time. Morphogenesis means the origin or genesis of forms. Forms means life forms. What we call basically making of an embryo. So we all animal and plant came from a single cell. That single cell divided, and then it made many cells. It made tissues, organs. There will be patterns in the organs, right? Like the stripes on a zebra and all. So those ideas, what we call the making of life forms or morphogenesis. And then he kind of get a nice elegant theory he proposed based on reaction diffusion. On the other hand, if you see the work of Max Delbruck, he was also a statistical physicist, and then he collaborated with a microbiologist, Salvador Luria, and he proposed many statistical models based on stochastic processes of Darwinian evolution in bacteria. John Hopfield just received Nobel Prize 2 years ago. From spin classes, he extrapolated that idea to artificial neural network. Right? By the way, in these few names, all these names which are in bold, this, and this, all are Nobel laureates. Okay? So, what I am trying to convey to you by biophysics, what we mean is not a narrow field. Is a diverse of topic where the ideas of physics, mostly statistical physics, in some cases quantum mechanics also can be applied, and even if you very original thinkers in physics, they were also very much motivated about this question of what is life. Even if you, many of you may know that Schrödinger himself, around towards his later career, wrote a book for What is Life, with mind and matter and autographical sketches. By the way, he was influenced very much by Max Delbrück, who was also a Nobel laureate, but in biology. Although he was a statistical physicist, he got the joint Nobel with his colleague and friend Luria. By the way, so you see, mind and matter is very interesting. What does it say? It says that Schrödinger is putting mind at the same bracket with the matter. So do you say that mind is also a kind of a very sophisticated form of matter? So this is the whole idea of this neural network that is coming out today. If you have a network of neurons that can have emergent properties, okay? So our mind, we can visualize also a form of matter. Anyway, so let's go forward. So what we do, we make models in biology, okay? And one of the very famous quotation of Charles Darwin, whom I believe to give the only theory in biology. So if you say that what is the most successful theory in biology, is of course the theory of evolution. Okay? Although you don't call that a theory like a Newton's law. Why? Because for a very long time of human history, there was no mathematical rules. What Darwin, told or taught us was mostly qualitative. But in later half, people like Ernst Mayr and also statisticians like Haldane and many people mathematized Darwin's theory. So he was one of the most successful theorists of biology, and what he is saying, all observations should be for and against some view if it is to be of any service. What simply means when you are observing something in the back of the mind, you are also having a concept, okay? And based on the concept you are observing. So you are not observing random things. You have a theoretical expectation already.
Test of hypothesis.
Test of hypothesis. Exactly. So this is how the science, all science work. You start with an observation, you hypothesize or conceptualize, which is basically an abstraction. Then you mathematically, formulate that, come up with prediction, and again it goes to an observation. So this loop goes on, right? And with this sort of motivation and sort of philosophy, this is basically what we do. So I will boil down everything into two topics that we are currently doing in our labs. One is we are trying to understand some subcellular biophysics. That means subcellular means happening inside a cell. Okay? And also tissue mechanics that is happening at the level of many cells. Okay? And we recently also started some simple experiments on bacterial population that I will not go into the deep on that. I will just slightly touch upon that. Okay, so far everything is understandable, right? We are talking in a very loose sort of philosophical way. But now we are going to be little concrete to take some examples of lab what we do. So the first topic is on subcellular level. So the life starts with what we call the DNA, right? So the DNA, as you know, it is a sort of a polymer, a biopolymer made with four kinds of monomer: A, C, T, and G. We call them in simplicity. There are long biological names, chemical names. I'm not getting into that. So you have A, C, T, G, and they are arranged in a long polymer. So imagine this as a kind of a segment of that DNA polymer, what we call a gene. Okay? And this arrow denotes that you are basically reading the gene from this side to that side. By the way, gene has a polarity. So both sides of a DNA is not the same. So when you are arranging the monomers from this side to that side versus that side to this side is not the same. So this arrow simply means that you are reading the code in the gene in one direction. So this scheme tells you what we call the central dogma of molecular biology. That what happens basically at the subcellular level. From a gene you make RNA, from RNA you make the protein, right? And the RNA can degrade, protein can degrade. Okay, so this is a very simplistic viewpoint how chemical processes of life starts. And protein is actually doing all the functions in our body. Okay, so all the diversity that we have is due to protein, like our eye color, our color of hair, our color of skin is basically some sort of one protein or set of proteins which are present in different abundance in different organisms. Okay, so this is what we call the diversity in phenotype. You may have all heard this word phenotype. Okay, from your class 12 biology. Phenotype simply means the characteristics of an organism. Okay, like I am bald. Okay, it is my phenotype. Okay, all of you have here. Many of you may not become even bald at the even old age. So having bald mustache like your HSR has nice hair. I am bald at this age. So these are like different phenotypes. Okay. So now the first part RNA from gene to RNA is called transcription. The second part is called translation. So first transcription happens, then translation happens. Okay. Now you imagine certain rates of these molecular reactions. You can visualize their molecular reactions. So Kr is the rate of making mRNA molecule from gene. Gr is the degradation rate of mRNA. Similarly, Kp is the rate of protein production. Gp is the rate of degradation of the proteins. Okay. Now suppose if you know this rate, then mathematically can we understand the number distribution or copy number distribution of these molecules, mRNA or protein. So to do that, we need to utilize a mathematical framework. Okay. So I am going to slowly describe that, and that's why I simplified the version. I have switched off the protein part. So we are only looking at the RNA level. Okay. So there is one RNA getting produced and that RNA is getting degraded. So we sort of can make mathematical inference of what will be the number distribution of these RNAs based on what we call writing down a chemical master equation. What is that? It's very simple that suppose I write N as the number of this RNA molecule at a time t inside a cell. Now, as you know that it will not be the same in every cells, right? So, if I think of a tissue, there are many cells. In all cells, I cannot say, suppose one RNA is present in say 30 copies. It is not going to happen, right? Some cell will have 20, some cell will 30, some cell 25. So, what you have, what you call cell to cell variation. That means you have a distribution of the RNA copy numbers. So this T and T is the probability of having n number of mRNAs at a time t. Okay. Now we can write a time evolution equation of this probability. What we call a master equation. How to write that? Which is very simple. We basically think of some gain in probability minus loss in probability. Okay. So how to do that? So schematically think that by degradation and synthesis, your number of molecules is getting changed by one unit, one integer, right? So it can go from n to n plus 1 or n minus 1 to n. How it can go? So from n minus 1 to n, it can go by like this arrow with a production. Similarly, with n plus n to n, we can go via a degradation. Now, if you are at a time t, if the number is n, it can become n minus 1 due to a degradation. N can become n plus 1 due to a production. Right? So now these arrows, these red arrows and green arrows denote what we call transition probabilities. Okay? From one state to another state, what is the probability of transition? Okay? And how you can construct those transition probabilities? You can construct those transition probabilities by simply looking at these rates. So imagine this red arrow first from n minus 1 to n. Okay. So what it says that you make one more RNA copy number. Suppose RNA copy number became from 2 to 3. When RNA is being produced, remember your gene is not getting destroyed. Gene is still there. The DNA segment is still there. Okay. So this is what we call a zeroth order production. So this production of RNA does not depend on how many copies of gene you have. Gene you always have one copy. So you have just a constant here. Okay. On the other hand, when you are degrading from n number of molecules. Okay. So degradation is proportional to the number of molecules present at a time. Yes.
Can you please repeat the part whether we are not dependent upon number of genes?
Please louder.
And please repeat the part whether we are not dependent upon the number of DNA fragments present.
Yes, that is simply because we have only one copies of gene. Suppose for any protein, in most cases, except few exceptions, there is only one copy of a gene. But that one copy keep on producing many copies of RNA. Okay, so always this is present at a single copy.
So this equation is not conserved.
This expression is not conserved. Yes. So now, however, you can make a steady state assumption. So now, on the other hand, when you are degrading, it is proportional to the instantaneous number. So you are degrading from n plus 1, you are having degradation rate times n plus 1. When you are degrading from n to n minus 1, it is gr times n, and this is again here. So now I basically collect this term together and write this equation. How you write it? You see, think of the first term. First term corresponds to this equation. So probability of going from n minus 1 to n, this is kr, times probability of being at n minus 1. So you are multiplying two probabilities here, right? This is what you call transition probability. This is what we call state probability or occupancy probability. Similarly, you also multiply two probabilities here. This is the transition probability from n plus 1 to n times probability of occupying the state n plus 1. So these are my gain terms. Similarly, I can write as the loss term. Okay. So this equation, I think, all sort of now intuitively understandable, right? If you have any question in this equation, ask me right now. So I'm moving forward. Okay. So next, what we do is solve the equation in the steady state. By solving, you will get this famous what we call a Poisson distribution. Okay. So what we do, I can, do a little bit more work to make you convince. So when I am taking steady state assumption, that means the cells have reached a state where the numbers of the RNAs are not changing over time on an average. Okay, this is what we call a steady state assumption. So the probability distribution has become steady. It is no more dependent on time. So this time derivative goes away. Okay. So if I put the time derivative to 0, what I will write is this kr times p of n minus 1. I'm omitting t. It is no more a function of t, right? Plus gr times n plus 1 times p of n plus 1 equals to kr plus gr times n p of n. So how do you solve this kind of equation? This is what we call a difference equation, right? So there are several ways of solving it. The first very intuitive way is that we will keep on putting values for n. N equals to say 0, 1, 2, 3, and from there we will try to guess a solution. This is what you call a method of induction in mathematics, right? So this is not always possible if you have a very complicated equation, but in this case it is possible. So what do you do? First of all, what are the values possible for n? N can take values, all integer values, right? Up to infinity. So then I start with n equals to 0. Okay? So first I put n equals to 0 in this equation. So there the first term is equal is 0. Why? If I put n is equal to 0, there is no meaning of a negative. Number of a molecule cannot be a negative. Right? So this term is 0. Second term will be gr. Here I am putting 1. Here I am putting 0. Here I am putting 0. So gr of p of 1 equals to kr. This part is 0 times p0. So I write p of 1 in terms of p of 0. Okay? So let me, the portion of the board is very small, so I am writing on the top. Okay? Is it visible from the back? So this is p of 1. Now I am doing p of 2. So to do that, I am putting n equals to 1 in this equation. So what will get? Kr times p of, now if I put 1, this is p of 0, this is valid. Okay? Plus g of r, this is 1, so it becomes 2 of gr, p of 2, right? Equals to kr plus gr, now this n equals to 1 times of p of 1, right? Now p of 1 is this, I will put this value over here. So you get k of r plus gr times this guy, k of r by gr times p of 0, and you will see there will be a cancellation. What is the first term? Kr square by gr. What is the second term? So that this gr will cancel, right? Kr of p of 0. Now you see this and this cancels from the both sides, right? So then this, sorry, p of 0 will be there. So now I can write p of 2 in terms of p of 0, right? So what is my p of 2? This is kr square by gr, and there was 2 gr. So this is half kr by gr whole square p of 0. Okay? Now if we keep on doing that, next term will be p of 3. Instead of half, you will have 3 into 2 into kr by gr whole cube, p of 0. You can do this as a homework, you can check. Okay, so if you keep on continuing, here you have successive multiplication, so it will be n factorial, right? And this will be kr by gr whole to the power n. So here luckily we have guessed the solution. This is what we call method of induction, right? So I write the full solution. I write the full solution as p of n as 1 over n factorial. Kr by gr. What is kr by gr? Is the production by degradation rate, right? This is the ratio to the power n p of 0. Okay? But now there is one problem, because p of 0 is a constant we don't know. So we have to determine that constant. Then your solution is full and final, right? Now p of 0, how do you calculate? Now utilizing what you call the normalization conditions, right? All the probabilities should be equal to 1. So if I sum over all possible n values, what is n values? Starting from 0 to infinity. This should be 1. And if I put it here, what is this? So suppose this ratio kr by gr, I'm giving a new name, say lambda. Lambda is a constant. So what you have? P of 0, lambda to the power n by n factorial. half over n. P of 0 is a constant, so out of the sum, right? Now this summation is a well-known series of exponential functions, right? So you can write this as P of 0 e raised to lambda, and this is equal to 1. So P of 0 is basically e raised to minus lambda. Okay? So you put it there. So instead of P of 0, you write e raised to minus lambda. So this is the solution of what we call this famous Poisson equation. This is a Poisson distribution. And Poisson distribution has a very nice mathematical property. What is that? Its mean and variance are the same. Okay? So this you can also check by summing. So this is your Poisson distribution, lambda to e power n, right? E raised to minus lambda. Now if I get average of n, which is defined as n times p of n, sum over all possible values of n. If you do that, you will get the answer lambda. So the average is basically ratio of the production and degradation, right? So since it is a ratio of production of degradation, it is very intuitive. The average is the balance between these two, how much you produce versus how much you degrade. But what is even more interesting and counterintuitive, if I calculate variance, it will be again lambda. So it has a peculiar property that variance by mean ratio is what we call a phenotypic. So what we have now understood, by checking the phenofactor, which is basically variation mean, you can understand if the production of a gene product, which is RNA, is deviating from Poisson or not. And this is a very interesting theoretical tool. So you need to now have this viewpoint that there are many cells and these things are RNAs. So there will be cell-to-cell variation of the RNAs. That means when I do a histogram, I will see something like this. Now this cell-to-cell variability in RNA, what we call noise of gene expression or gene expression noise. And if it is following a Poissonian dynamics, then the variance by mean ratio is 1. And that is not very much noisy. It will be a narrow bell-shaped curve, right? But in certain genes, the noise can be huge. It can be super Poissonian. What it means, if I calculate variance by mean, it can be much greater than 1. So depending on that, you can categorize the genes which are more noisy genes versus less noisy genes. Okay? And why biophysicists or biologists are interested in that at all? What is the implication for life? So here comes the implication, which is more interesting, I feel, because this noise creates this phenotypic heterogeneity. Right? We are all made of cells, but due to this molecular noise, some will be having more RNAs, some will be having less RNAs. So those people will have more RNAs will generate more protein. Some other people can generate less protein. So this create a kind of variation or heterogeneity, which is the main driver of evolution. So heterogeneity can come even at a single cell, not only human. Think of a single cell like a bacteria. Even there will be heterogeneity. Okay? So this will be a driver of evolution. So this is the hypothesis. That's why we are very much interested in this topic called gene expression noise, to understand that. Now there are more research going on, and our lab is also involved that. So what we told, it is just the tips of the iceberg. So this was the scenario. We start with a gene, it is producing RNA, right? So this is a low noise thing. But biology has more complicated ways of regulating things. So what in biology people have experimentally shown, genes often switch between active and inactive states. That means there are other molecules which are called repressor. They come and bind to genes. Then the genes don't express. They don't produce RNAs. So that is an inactive state. If the molecule, this green balls, falls apart, then again they will express. Now if you write down the equations for that, you can understand now there will be two equations. No more you can describe with a one equation, because one you have to write for the active state, another equation you have to write for the inactive state. And this switching rates will be a very important factor. The switching rate is basically a binding and unbinding rate of the precursor molecules, right? Now, when you do that, you will have a very interesting prediction that you will get a bimodal distribution. It's a very non-typical distribution that we encounter in equilibrium statistical physics. We most often encounter Bell set. Now, this bimodal distribution, what we call a high noise distribution. And people can, check this in real experiments. So, what they do in bacteria, they can color code certain proteins. So, some proteins will be attached to some fluorescent molecules. Those fluorescent molecules, when you shine laser on that, you can monitor the fluorescence intensity. So here you see in certain scenarios, there are many different, distinct fluorescent level. Here you have a homogeneous level. So this is inside this bacteria, this kind of switching is switched off by some genetic engineering. Inside this set of bacteria, this switching is happening. So in experiment, when you measure that, you can get this kind of bimodal distribution in protein intensities. Now, you are not measuring actual protein or RNA, rather you are measuring the intensity of these fluorescent molecules, which are proxy for the protein or RNA. So there you can get bimodal distribution. So these are testable. Yes.
What is triggered? Why repression is triggered in some place from no heterogeneity? Higher
No, here there is no repressor molecule, there is a repressor molecule.
Yeah. Why?
And this so this coming from simply this binding unbinding kinetics. If the binding unbinding kinetics is probabilistic, okay, it is everything is random process. So this production is a random process, degradation is random process. Similarly, binding and unbinding is also a random process.
So no chemistry is there. I mean, is there any chemical chemistry is playing any role over here?
No, of course, chemistry of course has a primary role because chemistry is primarily determining this binding affinity. If it is very tightly binded, then you will not have this phenomenon. Because so this is what we call a separation of rates or separation of time scale is necessary. So this binding unbinding, if it is only overwhelmed by binding, so you bind, you don't unbind very often. That's what we mean by tight binding. Then it will not happen, right? Then most cases the gene will be in the onset. Mostly will get production. But when you see bimodal, when binding and unbinding are equally sort of probable, and those are much slower than production. Then what happens? Suppose in one cell the gene is on, and it stays on for a very long time, and then it goes off. Then again it stays off for a very long time. Then you are sampling from the on as well as from the off. Okay, so then you are creating a heterogeneity in time, which is basically showing up spatial heterogeneity among many cells. Okay, so do you get the intuition? You have to, of course, when you do the math, it will come out. The intuition is that this binding unbinding kinetics has to be much slower compared to the production and the decay. Then you will get two separate modes, two separate subpopulations. Okay, so how much time we
got the higher levels. The mode in the higher mRNA as the higher spread is more.
believe that yes. This will also depend on those rate instance, like you can determine the width of each mode. You can express that as a function of the binding unbinding ratio. So this binding unbinding ratio gives you a time scale. On the other hand, the production decay. So you have to decouple the binding unbinding and production decay. So this is the whole physical picture. So anyway, so let me move forward. So we are getting into more realistic or more complicated regulation, what we call post-transcriptional regulation. What does it mean? That when you make a RNA, there are another set of molecules, what we call microRNA, in short miRNA. These microRNA are very small. They come and attach to the RNA and make a mRNA microRNA complex, and then they can degrade. Either they can degrade both of them, or only the mRNA degrades, the microRNA remains. So now if I include this kind of dynamics, things will be much more complicated, okay, as you can imagine. So again, this is ongoing project, and my student who is present there has done a couple of papers on that. So what we are asking that what are the effect of this microRNA kind of negative feedback in gene expression noise. So why you call a negative feedback? Because what happens, these proteins are coming from this say blue gene. So this is this old scenario. You make a RNA, and from RNA you make a protein. Now microRNA, they are also coming from another gene, right? So you have another gene, which is this red gene that produces a microRNA. MicroRNA don't make proteins. They are very small. They attach to RNA and degrades it. So now, what happens? This RNA, this microRNA producing gene can crosstalk with the protein. So if this protein comes and binds to the microRNA, it will produce more and more microRNA. If the protein does not come and bind to this red gene, it will produce less microRNA. In that sense, this is a kind of that on-off kind of switching that I was talking about before. Okay. Now what happens? This microRNA and protein can also bind and make a complex, and it can decay in two different ways. Either both can decay or only mRNA can decay. So as you can understand, it's a very complex circuit. So in this complex circuit, what do you do? If you write the master equation, this is such a bad looking equation. Okay. So when my student wrote it, I told him that I don't want to see you try to solve. If you cannot solve, we will do it numerically. Okay. But as you can understand, there are many variables now. You see, R is for RNA, B is for protein, S is for microRNA, C is for complex, mRNA and microRNA complex. So this is a 4-variable master equation. If someone can solve it mathematically, then it is well and good, at least in some approximation. Or what we do, we write down all possible reactions. But all these reactions are probabilistic. And what we do, call a kinetic Monte Carlo simulation. Then you can predict also many things. So this has been published. So one of the small, theoretical things that we can show that there is a threshold crossing behavior in the mean RNAs. What does it mean? Now this is very intuitive. Why there is a threshold crossing? So there is a competition between this RNA production and microRNA production. RNA production is KR. Okay, these blue ones. And microRNA production is by this what we call Ks and Ks0 from two different states. Now, if RNA production is very high, microRNAs cannot suppress them. It will have lot of RNA. If RNA production is lower than microRNA, you will have suppression. So you go from what we call a low RNA to very high RNA. So there is a transition kind of behavior, but it is not a phase transition. This is what we call a very reminiscent of a phase transition and non-equilibrium crossover. You cannot call it a transition. But this crossover thresholds can be analytically, you can calculate by doing some approximation and neglecting fluctuations. Okay? And then then what more interesting is that if you look at this threshold, near the threshold, what is happening? RNAs are going down, RNAs are increasing, microRNAs are going down. So one side of the threshold you have more and more RNA, less microRNA. Other side of threshold you have more microRNAs, less RNA. In between near the threshold, both of them are in comparable amount. Okay? And near the threshold, that's why you have a dip in the correlation, negative correlation. The negative correlation coefficient is going to a highest negative here. And also near the threshold, you see again a generation of this bimodal distribution. Away from the threshold, you have unimodal distribution. Near the threshold, you have the bimodal. Why? Again, both molecules are comparable. Sometimes they are attaching, sometimes they are dissociating. Okay? Sometimes they are getting silenced. The RNAs are being silenced by microRNA. Sometimes they are open. So this is creating cell-to-cell variation. But if you see over time in a single cell, what is happening? In a single cell, below or above a threshold, one process is dominating the other. So this is microRNA stochastic process and RNA stochastic process. One is dominating the other. Here, you are saying anticoagulation fluctuation like a burst. You have a burst of RNA, then it is suppressing. Then you have a burst of mRNA, then it is suppressing. So it is kind of a stochastic oscillatory kind of a nature.
And does anything else, affecting on this temperature or anything else?
Yes, it must have an effect, but they are kind of assume no temperature dependent. Of course, there will be isolated system. Or you can think since it is a bacteria, you are maintaining the bacteria in a particular temperature. So like when you make yogurt, you maintain a particular temperature. So since temperature is not changing,
In physics, this transition probability in quantum mechanics,
yes,
temperature is very important.
Exactly.
So over here, why this transition probabilities are, why this transition is triggering this transition?
So because temperature is a very complicated concept in biology, because we still don't understand how to calculate temperature. Because in active matter and all, there is no notion of temperature, but there is a notion of fluctuation. But of course, these are fluctuation of molecules. But then temperature will bring some extra Brownian effect. So anyway, so let's move forward. So in next few topics, I will very quickly gloss over and tell you, because the time is short. I have how much time? 10 minutes? No, not 10 minutes. It's ending at 5, right? Okay. So you will not mind if I take 5 minutes extra. Okay. So for next few minutes, what we will do, we will quickly gloss over many topics which can be tackled in the same mathematical or numerical framework. And then I will go to a new topic. So another topic, what we call stochastic number of organelles in a cell. So idea is very similar. As you know, the cell is, so this is a textbook picture in class 12. So when class 12 you are made to memorize all this, people fix out. Okay, that's why many of us hated biology, right? So it is a field of many different structures. These are called organelles. But what biologists can do, they can measure the number of organelles in each cell. Nowadays, the microscopy is so powerful. So here we are showing what you call Golgi bodies and peroxisome. These are two different organelles, and they are marked by some proteins, again fluorescent proteins. And you can count the number of them per cell. Okay? And again you see very nice, kind of distributions, probability distribution. And what is interesting, this is Golgi body, this is peroxisome. One is showing nice bell-shaped curve, similar like a person. Here it is showing like a very long tail distribution, very highly skewed distribution. This is look like a gamma distribution and like that. So how this kind of things are coming? So how cells are tuning the number or regulating the number of organelles in them? So what biologists have identified, there are 4 fundamental simplistic processes. One is what we call de novo synthesis. That means an organelle is getting produced for the first time from kind of a membrane. By the way, this membrane is what we call this endoplasmic reticulum in a cell. So this is like a membrane source from where the membrane bulge out and you make an organelle. Okay? So this is production and decay. Same like RNA production and decay. But you have 2 new processes, what we call fission and fusion. So two organelles can come together and fuse, and also they can break apart, what is called a fission. So then we can write down four reactions. This is proportional to the number. This is also proportional to a number. But here I am writing these as the rate of fusion times n into n minus 1. Why is that? Because this is binary fusion. Binary fusion means two organelles coming together and fusing and making a larger organelle. Now this binary fusion has a combinatorial factor of NC2. So if you ignore the half, this is n half of n times n minus 1. So that's why you have to multiply by this factor. Half you can absorb in this rate constant. Okay. So now these are formulation, what we call chemical kinetic formulation, right? But if I think all these processes as random probabilistic process, then again you can write down a mastery course, right? And similarly, if I forget about fission and fusion, okay, then this will be again the same equation as before, and it will be again Poisson. So without fusion, even the number distribution of organelle will also have a Poisson distribution. Okay? But if you have all of them, then again you can have bimodal distribution in certain parameters. I am not getting into details. Okay? So what we learn? We learn by doing this exercise that which organelles are dominated by which process. If you remember, for Golgi bodies, I showed you a bowl-shaped curve. That means fission and fusion is not taking much for them, right? It is dominated by synthesis and decay. If you look at mitochondria, biologically it is known that they are dominated by fission fusion. Production and decay happens, but with a much slower rate. So if you can solve the full model, you can get different limit of the model. So that we did in a couple of papers. So luckily, it was exactly solvable in some limits. Okay, so that's why we publish in some mathematical based journals. If you cannot publish it, we send it to physics journals. If you can publish it, a little more mathematical journals. Okay, so another type of things that we do in subcellular processes. So this is where we are also trying our luck with experiments. What we do, we try to understand this number regulation of plasmid in a bacteria. So if you don't remember your high school biology, not physics. What is a bacteria? Bacteria, if you have a rod-like bacteria carry this kind of circular DNA. These are extra DNA than the genome. The genomic DNA is red one. These are called plasmids. And what is interesting is that this plasmid also can have antibiotic resistance gene. So if you give antibiotic, bacteria don't die because of having some genes which are antibiotic resistant. So this plasmid provides some protection. On the other hand, having too many plasmid is problematic because you need a lot of energy. So this is kind of a, you need to make a balance. If you have too many plasmid, you have protection from antibiotic, but also you are costing energy. Okay? So the question is that to keep or not to keep the plasmid if you have low antibiotic. So if you have low antibiotic, you don't need much protection. But if you are at high antibiotic, you need protection. So then what we are not getting into experimental details. Let's skip that. So what we did is that we put basically a plasmid, inside that we put a green gene, a gene that is producing some green fluorescent protein, so that we can visualize it, track it. So what we saw that if you don't give any antibiotic, ampicillin antibiotic, everybody was green because they were having plasmid. But stochastically, they are kind of releasing the plasmid, they are not keeping it. So then what you see, if you look at the distribution, these are plasmid bearing cells. And this is real experimental data. So you see bimodal distribution is emerging when there is a subpopulation that is keeping the plasmid, even when it is releasing. as a whole from the population. So, you see, I am very fascinated about this bimodal distribution. I keep finding and seeing it at different things. Because bimodal distribution is not very common thing that we encounter in physics, right? But in biology, it comes again and again. Because biology loves to create diversity. Okay, if you kill diversity, it is bad. Okay, so now very quickly I will talk about this. I will take maybe another 15 minutes. This is what we call tissue mechanics projects, on which Tunni is one of our student, your student is working, and I have another student working in this. I already gave part of this talk in previous seminar here. I think if I reduce this part, so I wanted to tell more about the other part. So here, a cell is making a tissue. So here the strategy is that we want to understand this, right? So we will take the idea of a statistical mechanical sort of a framework where I visualize the cell like a mechanical object, and you put a lot of mechanical object with some interaction. What kind of collective properties it will show? So in a sense, it is a similar conception like a kinetic theory of gases, right? So to give an experimental background, so this guy is a zebrafish embryo. I showed you in the beginning. So this is the head, and this is the tail, and this circular thing is the yolk. Okay. Now this is an experiment published in Nature that showed that if I look at the cell in this part versus in this part, cells are moving in a very different manner. In this part, this blue part, cells are kind of not showing very diffusive dynamics. They are caged. They are kind of caged by the labels. So the MSD is kind of getting flattened, where the MSD over here is diffusive. MSD means mean square displacement. All of you, I think in the BSc of MSc level, you know what is the mean square displacement. Now, why it is happening? It is happening because people have shown that in this back part, there are a lot of one protein that we call cadherin. Cadherin is what we call an adhesion molecule. It brings adhesion between two cells. So cells are very adhesive. But near the tail, cells are not very adhesive. So you see, nature has made a very interesting way of like it is like making a sculpture from a clay. So this tip of this tail is growing forward. So purposefully it is kept in a state which is little more fluidic. But the back part of the tail is kind of a solidified. Right? Because there the tissue, cell movement inside the tissue is minimized. Okay? Now if you look at the single cell level, what happens? These are cadherin mutant. Mutant means they, in these cells, these adhesion molecules are not getting produced enough. Okay? So you see cells are getting kind of a circular shape, and there is a cell-cell gap in the tissue. Here cells are having nice geometrical shapes. Okay? So let's skip this part. So now this thing you can also see in culture of a cell, in 2D culture. So previous thing what I showed you, this is in an embryo. It is very, very complicated thing. Even to image a real embryo, a real organism is not a easy job. That's why they published Nature by discovering there is a solid-like to fluid-like transition in the tail of a developing zebrafish. Okay? But here it is a much simpler experiment. You have a bunch of a cells and you putting together, okay, on a petri dish. And what they see that over time, day 1 to day 3, this RMS velocity is going down. And why they are going down? Again, you see these green dots are these cadherin molecules, these adhesion molecules. And over time, you see the green dots were everywhere in the cytoplasm. Now they are coming at the cell periphery. That means the cell-cell junction is getting matured over time. They are becoming more and more adhesive over time. So you can think the adhesion is increasing over time. And that's why cells are kind of jamming. Okay? They cannot move much. So how do you understand this kind of phenomenon? Transition. Okay, from liquid-like state to solid-like state. Whether you can even call it a real phase transition in a physics language, that is also not sure. Okay. So how do we model this kind of stuff? We model this kind of thing by thinking cells as active Brownian particles. Okay. So what is an active Brownian particle? Even before that, I think this framework, I don't know, at the BSc level you know it, but in MSc you must be knowing what we call the writing a Langevin dynamics for a moving particle. Okay, so for a Brownian particle or a Brownian motion, suppose you have a pollen grain, the famous example of a Brownian motion, right? You have a fluid, inside that you have a pollen grain, which is like moving in a random way. Why it is moving? It is moving because there are, water molecules which are coming and randomly kicking this guy. So you have random collision forces. Okay? So Paul Langevin, he sort of suggested a very Gaussian description with this equation. What this part is mass into acceleration. Then you have a viscous drag minus viscous coefficient into velocity. Plus there is something called eta f. Now eta f is a what you call a random collision force. So this is a very funny equation. It is not a deterministic equation. Part of the equation is stochastic because v dot m, these are deterministic variable or deterministic parameter. However, eta is a random variable. So you cannot solve it in an ordinary way. So you need this is called a stochastic differential equation. Okay. So now there are some character of this force. Since they are kicking this particle from all sort of direction, the average on an average should be 0 because if you take the vector sum, it will cancel out, right? So the average of this random collision force is 0. But there is a correlation, but the correlation will fade away very quickly if I look at a very far away in time. So correlation at time t and time t prime will only exist at t equals to t prime. But if t not equals to t prime, there will be no correlation. So you express that by writing as a delta function, right? So delta function only exists when they are this both variable t and t prime equal to each other. Otherwise, it will not exist, right? It is a concept what we call a diffusion curve. Okay. Now you can make it even simpler. What we will take the overdam limit. What is the meaning of overdam limit? There is a very precise meaning. So suppose I divide this thing, this equation later I said is gamma, the viscous coefficient. So in the acceleration part, mv dot, you will have this guy, m by gamma. Now if you do a dimensional analysis of m by gamma, you will see this is mass, this has dimension of force by velocity, right? If you put that, it will have a dimension of time. This is a dimension of time. Now suppose I take typical values of m I can take say the mass of a bacteria or mass of a pollen grain. So m is of the order of say picogram. Gamma, if I think of that of a water, okay, if I put these values, you will see the time it will get is very small, of the order of 10 to the power minus 7 or 10 to the power minus 8. Okay. Now you will not have any resolution of microscope to even look at that time. There is no microscope that can resolve 10 to the power minus 7 or 10 to the power minus 8 second difference in motion. Okay, so with this logic, what we can do, we can ignore this term. And this is what you call overdamping. That means your viscosity is so high compared to mass, I am ignoring the acceleration term. So this becomes a very simple equation.
I mean, without this water, if you use oil, this water limit will be
Even much more
That time will be reduced.
time will reduce faster, much clearer. So by the way, remember one thing, we are transformed the equation, right? So this eta is not same as eta s. This is another noise. So that's why I'm introducing another diffusion constant d, which is not same as dB dimensional. But the net assumption is the same. The average is 0 and correlation is a delta function. Now, if you do the calculation, you have to use the delta function integration. I'm not getting into that. Okay? We can, if any of you interested, I can talk you after the talk. So now you can show this famous law of diffusion, which is mu square displacement is proportional to the time difference, 2d of dt. Now this scenario changes when we think what we call an active Brownian particle. So it is a very interesting paper by Univasu in 2018. If you want to understand what is the nature of a single active Brownian particle. What is the meaning of an active Brownian particle? The meaning is that instead of a pollen grain, if you think of a bacteria, you imagine this, tiny bacteria with flagella. In the beginning, I showed you. So bacteria can, move their tails or flagella and make a motion of themselves. This is what we call self-propulsion. So they utilize energy and they move by themselves. Cells are also like that. So they can generate their own motion. So instead of writing like this, I can write over the equation in different way. So suppose I write in 2D, I write it as a vector. So I write that r dot as some v0 time n cap. This v0, I call it a speed. Now this is where there is no physics, it is purely biology. There is no particle which is a passive particle can have its own speed without subjected by a force. However, by generating energy, a cell or a bacteria can generate its own speed. In that sense, we call it an active particle. Okay? Now, I am not putting randomness directly in this equation. Why? The source of randomness in previous equation, this is uniform random collision of the water particles. Here I am putting the randomness in the direction of this n cap. That means this direction of the speed is random over time. Okay? So if I write n cap, since it is a unit vector, as cos phi and sin phi, we'll add another equation of phi, okay, as this. Some constant which is the strength of the noise times eta. I call it say eta phi. Now this noise, that's why different from these two noises. This noise what we call a noise in angle or angular noise. And this dr is called rotational diffusion constant. So because of this equation is a actual stochastic or noisy equation, you have a immediate effect on earth. Okay? So this is the idea of an active Brownian particle. So in if I break in component in x dot and y dot, you have v dot cos phi, v dot sin phi, and you have phi dot. Now if you solve this equation, what is interesting? At long time, again you will get back the similar imagery behavior proportional to time. However, you will have an effective diffusion constant which is v0 square by 2dl. So this effective diffusion constant is square of the activity, square of the self-propulsion speed. So this is the background. This is you have to remember when we want to model anything using active Brownian particle. Okay, so with that in mind, by the way, I just wanted to put this very interesting quotation by another famous statistical physicist. A good theoretical model of a complex system should be like a good caricature. It should emphasize those features which are most important and should downplay the inessential details. So this is the idea of what we call post gaining. So when we are looking at cells, there is no point on thinking all these molecules, genes, RNA, DNA, everything is gone. What is the essential details? The essential details is that the cell should have some speed of its own, and there should be some pressure, intracellular pressure, and there should be some adhesion between the cells. So we are keeping these three ingredients. So one cell, very simply a connected bead and spring. There is a pressure inside. So you boil down writing two different parts. One coming from the pressure part and one coming from the spring tension, which is basically giving you a surface tension. Right? Then each of these balls, each of these particles, I can assume like a ABP, active boundary particle. And between two particles of one cell or another cell, there will be some adhesion and repulsion. Right? So I am having some cutoff, some linear adhesion and linear repulsion. Okay? These are not very complicated equations. It looks like complicated equation, but if I give you the slide, you will look carefully. These are like spring forces. You can imagine there are two different springs, one giving you repulsion, one giving you adhesion, not one spring. Okay? And then this will be an equation of motion. You have a body force plus interaction force. But then I add this guy, activity. So then the activity comes from writing this extra equation in red part. So each of these balls now having a speed direction, which is p cap. And the direction of the speed, if I call it theta, which was phi there, also have an equation with a noise. So now if you have this equation, it is now multi-particle thing. Imagine that one cell has many beads, and then to make a tissue you have many cells. So it is a very complicated, what we call a multi-particle many body system. It's very difficult to do it analytically unless you make a continuum theory. That we are currently will do in the future. I have a dream of that. Or even before that, we are just simulating it using Langevin dynamics. And let's not get into the parameter details. So you have two different behavior in high adhesion versus low adhesion. You see, in the high adhesion, this is the result of simulation. This cell is not moving much. It is caged by the neighbor, right? In the low adhesion, you will see there will be neighbor exchange. That's why I am aligning the red and green to emphasize that. You see the red and neighbors, right? After some point, the green will be neighbor, red will be separate. You see now green becomes neighbor. You started the video with the red as neighbors, then green becomes neighbor. So slowly there are turnover, slowly there are exchanges of the neighbor that is going on, and that's why if I look at individual trajectories, it looks diffusive and it looks very caged. So this looks like this experimental trajectories. Now how you can quantify that? Now you can quantify that by looking at the diffusion behavior by the MSD. So you see in the low adhesion, in this direction adhesion is increasing. In low adhesion, this is proportional to time. In high adhesion, it is flattening with zero slope. Now I can define a diffusion constant, right? How I can define a diffusion constant? By this equation got mixed up. I don't know why. Sorry about that. So diffusion constant, if I define, then I normalize by that diffusion constant by this diffusion constant of a single active dominant particle, which I already showed, V naught square by 3. Then I will get a normalized value of the diffusion constant in a cell of a cell in a tissue divided by the diffusion constant of a single dominant particle, active dominant particle. And these will serve as an order parameter, right? And you see it is going from high value to low value with addition. Similarly, if I see the initial exponent from 1, it is decreasing to very low value, right? So you see a clear transition from diffusive to very subdiffusive behavior. And at the same time, there are many funny things going on. So here I am color coding the distance covered by a cell. Okay? This is the diffusive behavior. Here you see very heterogeneous nature. That means distance covered by one cell is kind of not same is same as the adjacent cell. So they are covering many different distances. This part is near the transition, near the boundary of liquid and solid. Here what you see, some are very blue area. That means these guys are not moving. Some are very bright area. That means these are moving. So you have zone of highly motile cell coexisting with the zone of clustering of non-motile cell. Okay. So this what is called a glassy behavior. So this is the situation that I keep on telling to the students when we discuss glass. Is like when you are running or traveling in a very packed up train in Seattle station. I remember I used to come back when I was a student of Jadavpur from Seattle to Bandel. My hometown is Bandel. So there, when you first go into the board at train at a particular station, everybody is packed. So naturally, you will create cluster of non-motile particles. We are particles, okay? And then at every station, there is a little bit of reorganization. Some particles are getting out, some particles are entering. Then what will happen? You will have small clusters of this motile thing, maybe near the gate, not in the interior part.
So it is near the.
This is near the transition.
I think divergence of the links take up the size of it.
No, that's why. So we call it a glass transition. I don't think it is a true thermodynamic transition. So we are trying to talk. I have talked with Chandan Dasgupta, who is a famous glass transition in IISER. So many people think glass transitions are not true phase transition in a thermodynamic.
You don't have any other direction.
We don't have adapter there. All these things we are basically classifying the phases depending on a cutoff of the diffusion constant. Okay. But this glass transition basically you have to characterize through look at the displacement distribution and what are the peculiar features of the distribution. So this is a PRL paper. This is in a experimental colloid system. So in this experimental colloid system, we have also marked the particles with the mobilities. Okay, and you see this cluster. So these are called colloidal classes. Now, when you look at the vector field of the displacement in this clustering position, you see kind of swirling pattern.
Vortex.
A vortex kind of pattern. So you see some vectors are very tiny, some vectors are very large. So this is also what we call dynamic heterogeneity, right? So you have coexistence of highly motile and less motile zones. Now what is even more interesting, such pattern also can be found in experiment of cells. Okay?
So is there concentration gradient is there?
No, there is no concentration. This is fully packed. So the packing fraction is close to 1, okay, of the whole system. So there is no, these are not density dependent transition. Okay, in normal colloid, what happens? You can tune the density, but in a tissue, there is no meaning of a tuning of density. Our skin, if I think of tightly packed,
This is something like cell polarized.
exactly. So this is like a very interesting. It is not a density dependent glass transition. This is a density independent glass transition coming via some adhesive interaction or some other factor or activity. If I reduce activity, then also there can be glass transition. So how do we characterize this kind of thing? We characterize by defining something called alpha 2 value. This is a cumulant, higher order cumulant of the displacement distribution. So this is fourth order moment divided by 2 into second order moment minus 1. Now for a Gaussian distribution, this is exactly 0. For non-Gaussian, this is non-zero. So you see there is a peak behavior near this liquid solid transition. which is telling you this is a highly non-Gaussian distribution. And what is interesting, in the liquid solid transition regime, this in the liquid regime it is Gaussian, but here it has exponential tail. Okay? So again you see deviation from Gaussianity. So as I told you, any statistical physicist will be very much intrigued if you bring deviation from Gaussian. Because we are bored, seeing Gaussian everyday life again and again. So even in this, bimodal distribution I talked about, that was a nice example of deviation from Gaussian. These exponential tail distributions are also a kind of a deviation from Gaussianity. Okay? And now, while it is happening, to understand the actual physics, what we have to look is that at this time scale, we have looked the actual motion of particles. So here in the simple diffusion case, this is a nice diffusive right. Okay? But here, if I suppose say at this delta T star, this is a time scale when the cumulus is peaking. That means you are very, very away from the Gaussians. Here you will see a very different trajectory. You see this one particular cell is kind of caged and hopping in the same position. Then suddenly it kind of make an excursion and go to another position. So this is what we call a cage breaking event. So what is happening, again you go back to this train analogy. So your train is coming from Sealdah. Okay. Now in Barakpur you need to get down. From the Sealdah you are like sitting in a cluster nicely. Before Barakpur comes, you feel like, no, now I have to get down. And then you have to go and quiz yourself from other people and other cells and make your way. So this is what we call a cage breaking event. Yeah. So these kind of events are happening, but these kind of events are also very, very rare. Okay? So it is becoming more and more rarer as we are approaching the class. So what is happening is not actually a class transition. It is not actually a real transition. It is happening in this way. These cage breaking events are becoming more and more rare as we increase the adhesion. That's why the system as a whole is getting jammed. So this is the physics behind it. Okay. Anyway, we don't understand everything. Still, there are many projects going on in my lab, and there are again this kind of signature in colloidal glasses. Anyway, so I don't want to get into more details on that. There are other slides. I will sort of skip it, and we can, of course, for a physicist, it is good to make a phase diagram. So we made a phase diagram, but again, these are not actual phase transition. But we can set a cutoff on the diffusion constant, and we propose a diagram like this that can happen in the 3D space of adhesion. This is the rotational diffusion constant and the intracellular pressure. So in this 3D space, we can demarcate the solid and liquid zone. Okay. Anyway, so I will now end today. So these are my students. So these people are theory. These red people. They are here, and unfortunately, the experimental people did not come. Okay, so these are my lab members, and as you can see, by the way, I am wearing the same dress here as the picture. I did not notice that. So this is a lab picnic we had like 2 months ago, and my daughter also came today with me, but she has gone to visit Victoria. With her mom. So at the end, I want to tell you something. I have tried to explain many things, and I have tried to motivate you both from the physics side as well as the biology side. But now biophysics is a very dicey position. People say that we talk biology to physics and we talk physics to biology people, then nobody understands anything. And they think that we are doing something with it. So we are kind of cheaters. Okay, so people don't like us. On the other hand, if you think carefully, that it is also a very difficult and exciting field. Because in classical physics, people have done whatever has to be done. So it is kind of saturated. right? In classical physics, all the problems that are remaining are extremely challenging and extremely, of course, very much mathematically challenging as well as conceptually challenging. But if you think of biology, there is no mathematical theory. So, truly speaking, there is only one theory in biology, I told you, that is the laws of evolution. However, laws of mechanics, but whatever biological molecules are doing, it has to obey the laws of mechanics. It has to obey the laws of thermodynamics. But what is the implication of that in life processes? Those are very exciting questions, and nobody knows them. Okay, so this is the purpose of my lab. We are trying to do that with all students and a little bit of funding. And I think I have done more than almost 2 hours, right? Huh? I think almost 2 hours. So many people over there, I think, got very much bored and not understanding so far anything. So I'm going to stop now, and thank you for your patience.
We have time for one or two questions. Yeah. Well, lots of questions we can ask. So you can email me. One or two questions. If you have.
Yeah, yes, that's
background. But why is it conflict for Pn equal to 1. I go to y equal to 1. I don't know where it is. How it equal to 1.
you are talking about that Poisson distribution. Oh, it has nothing to do with biology. Total probability should be 1, right? So if you sum over all the probability, this is the loss of probability, right? The total summation of all probability should be 1. Okay, it is just a condition, normalization condition.
In biology, there is something more known as concepts. This biology is a physical concepts. So this is different with biology and physics.
So now that is the challenge. Yes. Here the matter is conscious. No, but that's why I showed you this name of the book, this Schrödinger book. But what Schrödinger is trying to say is what we call consciousness also should obey physical laws. But there comes this philosophical debate of the view of consciousness. Do you need a creator to regulate the consciousness, or it is fully explainable by physical laws? So this debate is because if you think that consciousness is nothing but manifestation of some very complex neural network, then consciousness also at one point of time humans should understand.
Because is now mentioned in that mechanical. It's there very much.
It can be. I know that he has a very complicated theory. I heard his talk in calculation. He came once, long time ago. So.
Artichu, any other questions? Yes. Actually, I have one question. Okay, so
তুমি বাংলাতেও কোশ্চেন করতে পারো if you want to, or পরে বাইরে গিয়ে কোশ্চেন করতে পারো।
So let us kind of thank you.